Alright, the next focus of these lectures will be the SIM media. Now I love this test because what this test allows us to do is to test for three distinct processes. The first thing we're going to test for is sulfur reduction. Second thing we're going to test for is indole production from the amino acid tryptophan. And then the third thing we could test for is motility. So now let's look at sulfur reduction in the SIM media. Here we can see, here's our control tube. What you can see, and it's going to be even more clear in the laboratory, is a nice black precipitate is produced in that tube. That's an indication then that H2S was produced and that H2S reacted with that ferrous sulfate. So that's sulfur reduction. Let's talk about indole production. When we look at the pathway to produce indole, what we see here is we have another amino acid called tryptophan. Now, tryptophan is a very unique amino acid due to the presence of this indole group. We have that 6 carbon ring fused to a 5-membered ring. Now, using the amino acid tryptophan, some bacteria can produce pyruvic acid. Well, why is pyruvic acid important? Well, just as we talked about on the last slide, pyruvic acid can be used in respiration. It can be used in fermentation to yield ATP. Now, in order to convert tryptophan to pyruvic acid, the enzyme tryptophanase both deaminates the amino acid, which means it produces ammonia, and then also removes that indole group. So if the organism has the ability to convert tryptophan to pyruvate, indole is often considered a byproduct of that process. So now we can look for the presence of indole using what we call Kovacs reagent. Through the use of Kovacs reagent, if indole is present in this reaction, a nice red compound is produced, and it's the presence of that red ring on the top of our SIM media that's an indication that tryptophan was catabolized. Remember, tryptophan being catabolized produces the indole, and therefore the presence of a red ring is an indication that it was tryptophan broken down. Let's look at the results. Here's a control tube. We can see here in the tube on the left a nice red ring after the addition of the Kovacs reagent is an indication that tryptophan was consumed. We can see here in the tube on the right that after the addition of Kovacs reagent, we see no color change. Therefore, the lack of color change is an indication that no tryptophan was consumed. The last process we can look at using SIM media is motility. Now remember, bacteria use flagella in their motility. If an organism is non-motile, the organism will only grow along the stab line. Now one thing to keep in mind that SIM media is a solid media. So we use a needle in order to inoculate this media. Well, we can actually see where the needle was inserted in this tube, right down the stab line here. What we can see is as that organism grew, it grew only in that area in which it was introduced. Therefore, that's an indication that that organism in tube B is non-motile. Now, if you look here in tube A, not only are cells growing where they were originally introduced, those cells have now irradiated from that original line. And what you notice in this SIM media is it's uniformly turbid. It's growing everywhere. Therefore, the organism in this tube is motile. Now, why would you use these series of tests? Sulfur reduction, indole production, motility. We often use it to differentiate amongst the enteric family of bacteria. So in your unknowns, if you think you have a gram-negative rod, and you've observed sugar fermentation in your unknown, You can use the SIM media then to differentiate amongst the E. coli, Enterobacter, Klebsiella, Salmonella, Proteus, Shigella, Serratia. All of those can be distinguished from one another using SIM media. Now, let's test ourselves. These are very nice tubes or pictures we've taken in the laboratory. What is the interpretation of the organism in the tube on the right? That's right. There are actually two results I can see in this tube. The first interpretation I can make is that hydrogen sulfide was produced. And the reason I can confidently say that is there is an abundance of that black precipitate below the surface of the SIM media. Remember, thiosulfate reduction is an anaerobic process, therefore you'll see it anaerobically. Now, you notice I also see the presence of a pink ring at the top. So the second interpretation was that indole was produced via tryptophan catabolism. Remember, to this tube, we added the Covox reagent. And as we added that Covox reagent, we see that pink ring was produced, an indication that indole was present. Let's go on to the next self-quiz. What's the interpretation of this tube? Well, I see two things. One is easy to see, one may be more difficult. The first thing I could see is clearly that indole was produced. We can see that nice red ring on the top after we added Covox, and that's an indication this organism can catabolize tryptophan. Now the other thing, and I'm going to have a slide next that may show it even clearer, but you can see that is turbid throughout that media. Therefore, that turbidity throughout that media indicates that the organism was also motile. So what you're starting to see in the SIM media, you have to look at a variety of things. You have to add Covox, look at the top for the presence of a red/pink ring. You need to look in the media below the surface to see if it's motile. And as we'll see in a second, you also have to look for hydrogen sulfide production. All right. Now let's look at motility a little bit clearer. I like this picture. What is the interpretation of this SIM tube? Well, I have a few interpretations. The first thing you notice is that there's only growth along the stab line. We did not see the movement of that organism throughout the media. Therefore, the first interpretation was that the organism is non-motile. We've also added Covox reagent. We can see now in the tube there is no red ring, therefore there was no tryptophan catabolism. The other thing you notice is there's no black precipitate. Therefore, we are confident that no hydrogen sulfide was produced.